Cell and tissues.
In this video, you will learn microscope and emergence of the cell theory, cellular structure and functions.
Light microscope.
A light microscope works by passing visible light through a specimen. One lens produces an enlarged image of the specimen, and the second lens magnifies the image and projects it into the viewer's eyes or onto the photographic film. A photograph taken through a microscope is called a micrograph. A light microscope and magnification only about 1500 times without causing barriers. Its magnification is 1500 times larger than the actual size.
Electron microscope.
Electron microscope is the most advanced form of microscope. In electron microscope, objects and lens are placed in a vacuum chamber and a beam of electron is passed through the object. Electromagnetic lens enlarge and focus the image on a screen of photographic film. Electron microscope can magnify objects about 250,000 times larger than the actual size of the object.
Cellular structure and functions. Number 1. Cell wall.
The outermost layer of the plant cell is known as cell wall. Cell wall is a non-living and strong component of the cell located outside plasma membrane. It provides shape, strength, protection, and support to the inner living matter of the cell. The outermost layer of the plant cell is known as primary cell wall, and the cellulose is the most common chemical in it. Some plant cells, for example the xylem cells, also have secondary cell wall on the inner side of the primary wall. There are many pores present on the cell wall. These pores are called plasmodesmata.
Cell membrane.
All prokaryotic and eukaryotic cells have a thin and elastic cell membrane covering the cytoplasm. Cell membrane functions as a semi-permeable barrier, allowing a very few molecules across it, while fencing the majority of chemicals inside the cell. In this way, the cell membrane maintains the internal composition of the cell. Cell membrane is mainly composed of proteins and lipids, with small quantities of carbohydrates. The electron microscope examination of the cell membranes have led to the development of fluid mosaic model of the cell membrane. The fluid mosaic model, there is a lipid bilayer in which the protein molecules are embedded. The lipid bilayers give fluidity and elasticity to the cell membrane. Small amount of carbohydrates are also found in the cell membrane. These are joined with the protein or a lipid of the membrane, and eukaryotic cell cholesterol is also present in lipid bilayer.
Cytoplasm.
Cytoplasm is the semi-viscous and semi-transparent substance between plasma membrane and nuclear envelope. Inside the cytoplasm, various cellular organelles are present, which perform various cellular functions.
Cytoskeleton.
The cytoskeleton is a network of microfilaments and microtubules. Microtubules are made of tubulin protein and are used by a cell to hold their shape. Microfilaments are thinner and are made of actin protein. They help cells to change their shapes.
Nucleus.
A prominent nucleus occurs in eukaryotic cells. In animal cell, it is present in the center of the cell. Nucleus is bonded by a double membrane known as nuclear envelope. Nuclear envelope contain many small pores that enable to act as semi-permeable membrane. Inside nuclear envelope, a granular fluid, nucleoplasm, is present. The nucleus is a dark spot and it is the site where ribosomal RNA are formed and assembled by ribosomes. Chromosomes are visible only during cell division, while at the interface of the cell, they are not in the form of the fine thread-like structure known as chromatin.
Ribosomes.
Ribosomes are tiny granular structures that are either floating freely in cytoplasm or are bonded to endoplasmic reticulum. Each ribosome is made up of almost equal amount of proteins and ribosomal RNA. Ribosomes are not bound by membranes and so are also found in prokaryotes. Eukaryotic ribosomes are slightly larger than prokaryotic ones. Ribosomes are the site of protein synthesis.
Mitochondria.
Mitochondria, the singular is called mitochondrion, are double membrane bonded structures. They are found only in eukaryotic cells. These are the sites of aerobic respiration and are major energy production centers. The outer membrane of a mitochondrion is smooth, but the inner membrane forms many enfolding called cristae. In the inner mitochondrial matrix, this serves to increase the surface area of inner membrane on which membrane-bonded reaction can take place. Mitochondria have their own DNA and ribosomes. The ribosomes of mitochondria are more similar to bacterial ribosomes than to eukaryotic ribosomes.
Plastids.
Plastids are membrane-bound organelles that occur only in plant cells. There are three types of plastids: chloroplast, leucoplast, and chromoplast. Chloroplast is bound by a double membrane. The outer membrane is smooth, while the inner membrane gives rise to sac-like structure called thylakoid. The stack of the thylakoid is called granum, while its plural is called grana. Grana float in the inner fluid of chloroplast stroma. Chloroplasts are the sites of photosynthesis in eukaryotes. They contain chlorophyll, mean the green pigment necessary for photosynthesis, and associated pigments. These pigments are present in thylakoids of grana. The second type of plastid in plant cells are chromoplasts. They contain pigments associated with bright colors and are present in the cells of flowering plants and fruits. Leucoplasts are the third type of plastid. They are colorless and store starch, proteins, and lipids. They are present in the cell of those plants where food is stored.
Endoplasmic reticulum.
The endoplasmic reticulum is a network of interconnected channels that extend from cell membrane to nuclear envelope. The network exists in two forms.
Number 1. Rough endoplasmic reticulum.
Rough endoplasmic reticulum because of its rough appearance due to numerous ribosomes that are attached on it. Rough endoplasmic reticulum serve as a function of protein synthesis.
Number 2. Smooth endoplasmic reticulum.
The smooth endoplasmic reticulum is involved in lipid metabolism and in the transport of material from one part of the cell to the other part. It also detoxifies the harmful chemicals that have entered the cell.
Golgi apparatus.
Golgi apparatus are set of flattened sacs-like structure called cisternae in the cell. In this set, many cisternae are stacked over each other. The complete set of the cisternae is called Golgi apparatus or Golgi complex. It is found in both plant cell and animal cell. It modified molecules coming from rough endoplasmic reticulum and packed them into small membrane-bonded structures called Golgi vesicles. These sacs can be transported to various locations in the cell or it to the exterior in the form of the secretions.
Lysosomes.
Lysosomes are single membrane bonded organelles. Lysosomes contain strong digestive enzymes and work for the breakdown or digestion of food and waste material within the cell. During its function, a lysosome fuses with the vacuoles that contain the targeted material and its enzymes break down the material.
Centrioles.
Animals and many unicellular organisms have hollow cylindrical organelles known as centrioles. Each centriole is made up of 9 triplet of microtubules made up of tubulin protein. Animal cells have 2 centrioles located near the exterior surface of the nucleus. The 2 centrioles are collectively called as centrosomes. Their function is to help in the formation of spindle fibers during cell division.
Vacuoles.
Vacuoles are fluid-filled, single membrane bonded organelles. Cells have many small vacuoles in their cytoplasm. When a plant cell matures, its small vacuoles absorb water and fuse to form a single large vacuole in the center. Many cells take in minerals from outside in the form of food vacuole and then digest the material with the help of lysosomes. Some unicellular organisms used contractile vacuoles for the elimination of waste from their bodies, for example, amoeba. Thanks for watching. Please like, share and subscribe my YouTube channel.